Hello, my name is Melina. I've been working on this project for a couple of months now with my mentor. I have decided to title it Exploring the Potential of CRISPR-Cas9 in Targeting the Genetics of Aging in order to extend human lifespan. So, this project is going to be investigating gene editing approaches to delay aging and enhancing longevity. Okay, so we're going to be understanding aging first and foremost before getting into the processes and genetically modifying the aging process. So, what is the definition of aging? Aging is the time-related biological process of becoming older, which is characterized by the deterioration of functions necessary to live. While aging is considered a natural process, various factors do actually influence how one ages, and it contributes to age-related diseases such as obesity and cardiovascular diseases. So, what are the causes of aging? These include internal and external factors. External factors such as socioeconomic conditions, the economy in which you live in, the society in which you live in. Internal factors include genetic diversity and immune system responses. So, what are the hallmarks of aging? Scientists have discovered many processes in the body that actually contribute greatly to aging. These hallmarks include DNA instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulating nutrient sensing, mitochondrial dysfunction, stem cell exhaustion, altered intracellular communication, cellular senescence, and chronic inflammation. Each hallmark has a specific process that contributes to aging, and although all the hallmarks are significant when it comes to aging, some hallmarks subject has increased in focus, such as cellular senescence and chronic inflammation. So, what are these hallmarks? Cellular senescence involves the permanent stop of the growth of individual cells due to mutation or damage within the cell, and it is called a irreversible growth arrest. And when these senescent cells has become established, they release pro-inflammatory molecules which trigger what is known as chronic inflammation, also called inflammaging or inflammaging, and it is a prolonged immune response that further damages tissues through inflammation. So, how do these impact the cellular functionality? Aging cells exhibit decreased functionality, which impacts tissue regeneration, immune response, and metabolic processes. It declines the cellular performance, which contributes to age-related diseases, and it reduces the adaptability to stress, which overall deteriorates the health of an aging person. So, what is the role of genetics in aging? Genetics plays a pivotal role in aging. It influences the rate of cellular senescence and other major hallmarks, which we've just talked about. Researchers have found over 20 genes, such as the genes that we're about to discuss right now that actually are involved in aging-related processes such as oxidative stress resistance, DNA repair, and telomere shortening. These factors lead to dysfunction and eventual cell death, which overall contributes to the aging process. So now that we've discovered what aging actually is, we're going to discover the genes that contribute to aging. So these genes actually include IL-11, Cat7, OCT4, and P53. They all play a role in aging by influencing various cellular functions, which we've mentioned before, such as DNA repair, protein synthesis, and cell division. So one of the first genes that I mentioned was IL-11. So what is IL-11? Interleukin 11, which is short for IL-11, is a vital cytokine that plays a significant role in managing the immune system. It regulates inflammatory responses, so it can influence the results of many diseases, which include age-related diseases. It possesses a double role in inflammation, which depends on the situation. It could either encourage or hinder inflammatory responses. For instance, in some situations, it may lower severe inflammation, which encourages tissue repair and wound healing, which is good. But if it is dysregulated, as frequently in the case with diseases that are age-related, like obesity and cardiovascular diseases, it can lead to chronic inflammation, which we have mentioned is a hallmark of aging. One critical way that the gene affects aging is by promoting cardiac fibrosis and remodeling. So, cardiac fibrosis occurs when scar tissues form in the heart, and it makes it stiff and harder for blood to pump effectively throughout the veins, which it arises due to a variety of factors, such as inflammation. And it is obviously more common in the elderly. So IL-11 in this case, it functions as a profibrotic cytokine, which means that it actively promotes the process of creating scar tissue, leading to the development of fibrosis. So the development of cardiac fibrosis is a key factor in the deterioration of heart function, which is observed in older adults. Another gene is the lysine acetyltransferase 7, which is known as CAT7. It has a primary function of adding acetyl groups to certain parts of histone proteins in order to open up that DNA structure and help regulate gene expression. It is crucial for several key processes such as cell cycle function, the stem cell maintenance, which is the maintenance of the versatility of stem cells, which it enables cells to turn into various cell types, and DNA repair. So the levels of Cat7 actually vary during the aging process, and it affects its function. One big effect of aging is the declining levels of Cat7, which leads to less histone acetylation. Histone acetylation is a critical process that makes gene transcription more accessible. So when Cat7 levels drop, it is accompanied by the reduction of active transcription of genes, which is necessary to maintain stem cells. So this decline is significant because in the context of aging, stem cells are necessary for tissue repair and regeneration. Another major consequence is the loss of cellular flexibility. Over time, cells are unable to adapt and effectively respond to signals, which leads to impaired tissue repair. And the inability to adapt can lead to inflammation or accumulation, which is two of the major hallmarks that we've discussed. And over time, it eats away at the efficiency of tissue repair, and it obviously exacerbates the aging process. Additionally, studies also suggested that a decrease in Cat7 functionality can begin or even worsen the cellular senescence process, which thereby promotes the overall aging process. So it is a very bad gene. Moving on to OCT4, octamer binding transcription factor 4, short for OCT4, is a protein that plays a critical role in the development and maintenance of pluripotent stem cells, which have the ability to develop into any type of stem cell, any type of cell in the body, which we mentioned before, a stem cell. In addition to its role with stem cells, it is vital for embryonic development. It forms a group of cells within the embryo called the inner cell mass. So, aging causes significant changes in the way genes are expressed due to epigenetic alterations. These changes can impact OCT4 in two ways. It can decrease the levels of OCT4 expression, which weakens stem cell identity, or it could cause abnormal changes in OCT4, which leads to cells becoming more differentiated. Epigenetics can actually silence OCT4, and when these levels of OCT4 drop, stem cells struggle to maintain their identity. So not only do epigenetic changes affect OCT4, but so does oxidative stress. Oxidative stress is when there is too much damage caused by harmful molecules called reactive oxygen species, also known as ROS, and antioxidants cannot fix this damage fast enough to avoid further damage. High levels of ROS can damage cellular components and disrupt normal functions, including the function of OCT4, thereby reduces the ability of OX4 to maintain stem cells, which then results in less effective tissue regeneration because we need those stem cells to effectively repair tissues. Last but not least, we have the gene protein P53. The tumor protein P53 is a protein that regulates cell division and death. And most importantly, it prevents tumor formation. It plays important roles in cell cycle arrest, cellular senescence, and differentiation in response to various cellular stressors, such as oxidative stress, which we just mentioned. So P53 is often referred to as the guardian of the genome. It has the ability to preserve genetic material in response to sensing cellular stressors, such as DNA damage. When it detects damage, it initiates a series of cellular responses aimed at either preventing the damage or preventing damage in other cells, which, as we know, accumulation of bad genetics leads to cancerous cells, cancer. P53 functions primarily at two checkpoints in the cell cycle. The first one is in between the G1 phase and the S phase. Cells prepare to replicate their DNA. So, if there is damage detected by P53, it will begin cell cycle arrest, also known as cellular senescence, which we've mentioned is a major hallmark of aging. It pauses the cell cycle right before it enters their S phase, where DNA begins to replicate to prevent further damage. So the other make checkpoint is between the G2 and the M phase, where the cell is ready to leave its G2 phase and move on to its mitosis phase. Similarly, P53 halts the cycle if damage is shown, giving cells any time to complete any repairs before attempting their division. If the damage is considered irreparable, it activates cell death to prevent increase in the number of cells with damage, which is also known as potential cancerous cells. The P53 protein is a crucial regulator of cellular senescence as it stops the growth in response to stress and damage. Over time, the effectiveness of this protein reduces in response to stressors, which leads to reduced regulation of the cell cycle and less effective DNA repairing processes. The decline of P53 can increase the amount of cells with DNA damage produced, which increases the risk of accumulating detrimental mutation and acquiring diseases, which is very, very bad. So it is good at the same time it is bad. So now that we've learned about the related genes, we're gonna learn about what it is we're going to be using to target these genes, also known as CRISPR-Cas9 technology. So, what is the history of the CRISPR-Cas9 discovery? CRISPR technology and its gene editing capabilities were discovered in 2012 by scientists Jennifer Doudna and Emanuela Charpenter, marking a milestone in biotechnology. Very significant discovery. It is short for clustered, regularly interspaced, short palindromic repeats in combination with the CRISPR-associated protein Cas9, which is a gene editor that can be used to modify DNA of various living organisms. So, how does this CRISPR-Cas9 technology work? So, it functions through a simple mechanism involving guide RNA that directs the Cas9 enzyme to a specific DNA sequence. As we can see in this picture here to the left, we have the yellow guide RNA, which leads this big blue blob, which is known as Cas9. It leads it to the specific DNA sequence where once it is detected, Cas9, which is also known as these molecular scissors, it cuts the DNA strands and it initiates the cell's repair process to alter the genetic material, whether it is adding, taking away, or replacing this genetic material. So, using CRISPR to edit these genes, the emergence of CRISPR technology has opened a new frontier in the field of genetic research, especially for age-related genes such as IL-11, Cat7, OCT4, and P53, which were our four key related genes. So, how are we using CRISPR specifically to target these genes? Starting off with IL-11, two approaches to slow down the aging process would involve either the inhibition of IL-11 signaling or the complete knockout of IL-11. So, IL-11 signals through a receptor called glycoprotein 130. It activates various pathways essential in IL-11 signaling, such as the STAT3 pathway. So, using CRISPR technology, researchers could disrupt these transcription factors, such as STAT3, in order to interfere with the signaling and reducing inflammation. Another way is CRISPR interference, also known as CRISPR-I. It is a technique that uses the CRISPR system to reduce but not completely eliminate the activity of a gene. It could be used to knock down specific genes involved in the signaling pathway, which diminishes IL-11 effects, but it doesn't affect the gene itself permanently. Alternatively, completely stopping the production of IL-11 could potentially reduce its contribution to chronic inflammation, which accelerates aging. Although the benefits of modifying the IL-11 gene include reducing the risk of fibrosis, cardiovascular diseases, chronic inflammation, and increasing cellular regeneration, potential risks include the infection of the immune system and the effects on other signaling pathways that have nothing to do with IL-11. Moving on to Cat7, it has a similar approach to IL-11. We could use CRISPR-Cas9 to eventually knock out this gene. So by disrupting the Cat7 gene, could help cells act younger by stopping the usual processes as a person ages. So removing Cat7 in um, older HMPCs, which is a type of cell which is found in the bone marrow and other tissues, it can slow down the aging process or delay the onset of cellular senescence. CRISPR-I, as we've mentioned before, CRISPR interference, could also be used to reduce its expression, which turns down the Cat7 gene, which reduces how drastically chromatin is remodeled. This affects gene expression greatly, and it keeps DNA packaging processes stable. So by disrupting Cat7, cellular senescence may be alleviated or even reversed. Stem cell function and regeneration could improve, slowing down the speed at which tissues age. However, over-inhibition of Cat7 could affect the ability of cells to respond to different stresses or signals over time. So an approach like this would need to make sure that it is very, very carefully balanced. Moving on to OCT4, we have a little bit of a different approach in this way. We're going to be looking at the actual activation of this gene, which is known as CRISPR-A, CRISPR activation. It is a technique that uses the modified version of CRISPR-Cas9 to actually increase the expression of these specific genes without changing the DNA. So it is basically like knocking out the gene or interfering with the gene, but the complete opposite, just activating the gene. So this approach would potentially rejuvenate cells, which restores their ability to function properly and making them more stem cell-like. An increase in stem cells could actually enhance tissue regeneration and improve how age-related damage is repaired because there are more cells that are able to turn into beneficial cell types. On the other hand, using previous methods, knocking out OCT4 completely may result in the loss of self-renewal in stem cells, which could actually harm tissue regeneration. So the activation of OCT4 could help revive cells by making them more like stem cells, which improves repair and regeneration. This method had actually been tested in animals to treat age-related diseases, such as the Hutchinson-Gilford progeria syndrome. However, the overexpression of OCT4, once again, could raise the risk of cancer by directly increasing cell accumulation, by directly increasing those amount of stem cells, which could be bad and it could be good at the same time. Last but not least, P53, a potential way it could involve the knocking out of the gene in order to slow down aging by preventing cellular senescence. The knockout could minimize the accumulation of cells. However, if it were to be completely stopped, it would no longer be able to prevent or detect damage within the cells. So, conversely, using CRISPR-A, CRISPR activation, it could include cell cycle arrest and DNA repair in response to stress. Increased activity, um, increased activity of P53 could help clear damaged cells with accumulate with age. Rather than fully knocking out or activating the P53 protein, a more nuanced approach would actually go back to our IL-11, CRISPR-I, CRISPR interference, which regulates p53 activity. This would reduce harmful effects while still allowing for that effective stress responses to maintain, also to maintain tumor suppression. So overall, p53 inhibition could potentially extend cellular lifespan by preventing senescence, but it could also raise the risk of genomic instability or eventually cancer. So we've looked at potential uses of CRISPR in these genes. Now we're going to be looking at what are the actual uses of CRISPR that scientists are currently applying now. So many studies have demonstrated its use in therapeutic treatments that modify the genome in order to actually improve healthcare outcomes. So applying CRISPR to genomic studies can aid in the creation of engineered animal models, enhance research, and the understanding of human diseases, which is good. CRISPR-based therapies offer a precise way to alter DNA, which actually target the root of the disease and potentially come up with a permanent treatment. Whereas traditional drugs, they manage systems when attempting to treat a disease. So its ease of use have made it a preferred choice for many genetic engineering projects. What are the future prospects in aging research? So the potential of CRISPR-Cas9 in aging is very vast. It has so many ongoing studies going on, identifying genes related to lifespan. CRISPR, by actually identifying and targeting these genetic factors, CRISPR may offer solutions to delay aging processes and even hopefully improve healthcare and improve the lifespan of a human being. And that is what we hope. Thank you and have a good day.